Now we move on to another important topic and an element of science that can be quite fascinating. Dr. Lundy Richards and Mr. Jonathan Smellie will be presenting on genomic sequencing for cancer diagnosis and therapy. Dr. Lundy Richards is the consultant medical hematologist, oncologist, and Jonathan Smellie is the principal molecular biologist for the Cayman Islands Health Services Authority. We do invite you, gentlemen, to come on up for your presentations. Let's welcome them. Just making sure we have the clicker all sorted.
Is there a laser pointer?
Pardon?
Is it laser pointer? Yes?
Yeah.
Okay, cool. Okay. Morning, everybody. Sorry to introduce a topic like this before 10 a.m. My name is Jonathan Smellie. I'm the principal molecular biologist and co-founder of the Cayman Islands Molecular Biology Lab. And I'm here with Dr. Lundy Richards, consultant hematologist and specialist oncologist, to discuss the implementation of cancer genetic locally in the Cayman Islands for the first time. So let's get started. Right here. Okay. All right. So to kind of ease us into this, this is a kind of graphical representation of the entire genome, right? And since about 2001, this is really what we've been interested in studying. So you have here all 23 chromosomes, and within those chromosomes, if you get a bit deeper, you have nucleotides, which are the individual letters, the A's, T's, C's, and G's that everybody is familiar with. So in our genome, we have 3 billion of those letters, and between each and every single one of us, we have a difference of about 4 to 5 million letters. So that's what makes up the variation that you tend to see between people, so your height, your hair color, all those different phenotypic characteristics that we easily identify. And those things can also be related to our clinical outcome as well. So that's why we're interested in studying those particular differences. So to go a bit granular, those differences are called in our field a single nucleotide polymorphism, right? And single, meaning the individual base letter, nucleotide, of course, and then polymorphism, a difference. And you want to look for ones that are common within certain subgroups of the population, depending on the disease. So we have common single nucleotide polymorphisms, which are ones that have at least one per 1,000 people in the population. And it's greater than frequency in that population than 1%. So those are not necessarily implicated in the disease. You can assume that because they're in healthy people as well. They're really highly represented in healthy people. And then if you're looking at a subset of individuals with a particular condition, and then you're able to pull out single nucleotide polymorphisms that are associated with that condition potentially, that's what we would call a variant that's associated with that condition. So what are the different types of variant? The different types, these are just 4, which are somewhat digestible. So there's a missense mutation. So you have your base pairs at the top there, your nucleotides, and they're read in codes of 3. And those 3 create amino acids, and those chains of amino acids are put together to make proteins. And proteins are really the functional components that drive all the different processes within your cell. And so you have here at the top, which is what you'd consider like the wild type or the reference, and you see that it creates these different histone proteins, and then you have a change in those nucleotides that leads to a different protein being, or amino acid rather, being made. And that's a missense. You have a deletion where you'd have a removal of that nucleotide, and then those 3s are read differently. So all the things downstream from that are different, all of those different amino acids, right? You have an insertion, which is what the name suggests. You have an insertion of a different letter, and then that also changes the way in which amino acids are read. Then you have nonsense mutations. So you'd have perhaps a change that tells the enzyme that's reading that DNA to stop reading and just create the protein at that point. And that can lead to a shortened protein, and that's generally something that's implicated in disease, because if you have a shortened protein, it's not functional. If it's not functional, it's going to probably create problems. So what is cancer, and how does cancer relate to what I just spoke about? Well, cancer is actually a genetic disease, and this diagram really is the result of a lot of very impressive work by Hanahan and Weinberg, published in Nature, and it shows all the different genetic components to the development of cancer. So you need all of these things to be happening for a cancer to start, and that's why cancer is such a complicated disease to treat and understand. So it's caused by the changes in your genes which control the way your cells grow and multiply, and you'd see here some of the easier to understand ones are growth suppressors. So there are certain built-in functions in your cells that prevent those cells from growing uncontrollably. And when there are mutations introduced in those processes, then they break down, and they're no longer suppressing the growth of those cells. And then you have immune destruction. So generally, immune cells are really good at identifying cancer cells. But in certain cases, when the profile of that cell may change due to mutations, the immune cell won't recognize it anymore and won't get rid of it. Then you have genomic instability, mutation. So for example, you have ultraviolet light that creates damage in your DNA, and the way in which that damage is repaired may be incorrect, and that will introduce mutations and cause those problems. Right, so you can see that cancer is a really hard thing to tackle because there are a lot of things going on here. So kind of to come back out a little bit and reframe it, what is genomics actually? So genomics is the study of the entire set of genetic information from an organism, right? So all of the letters, all of the chromosomes in the individual or person. And so genetics is the study of a subset of genes that then go on to perhaps be inherited, and there's the individual composition and how they actually work in particular processes that you're interested in. So genetics has been around for a long time, and genomics has really only started from the completion of the Human Genome Project, 2001, and all of this detail that we're unpacking about different diseases and correlations and risks and so on have come from that work. So how does that actually happen? Like, how do you even start that process? And really, it starts with, ideally, blood. You get a blood sample from a patient, and then you want to purify the DNA from that, and then you start the process of preparing it for sequencing. So that there is actually a sequencer. It's an Illumina MiSeq. It's an instrument we have in the lab, and that is used as a gold standard for clinical sequencing. And so once you put it onto that instrument, it can run for days in a lot of cases, and it's going to produce a huge amount of information about your genome. And that information looks like this. So once the instrument is satisfied that, a T or a C or an A or a G is where it says it is, right? That's that quality assurance aspect. You then have to align all of these different things, because the genome is read and then sort of read into chunks, and then you have to stitch them together. And to make sense of that, you take you download data from the national databases that would have the reference of what you're putting this alignment to, and then you can say, okay, this part of the genome is here, and this part is here, and so on. So you're able to compile the actual genome that you've sequenced. And once you do that, you then look at the differences. So I mentioned before, you have 4 to 5 million differences between individuals, right? And that's what we're really interested in. So those are called variants, and variants are put into a file, and then that's your end result, your variant call file. So that's just the second step. The third is that you have to interpret this now. So this is a very, very rigorous process, and it generally involves a lot of clinicians and a lot of scientists, and you look and interrogate genetic databases, literature and research, and do statistics and modeling, and your phenotypic data from your medical healthcare records. And the idea is that you can look and see trends and say, okay, we have this individual, they present with this particular disease, and they have these mutations. Now, is this something that a group in the UK or Australia or Canada are also seeing? Is there a relationship there? Remember I was saying earlier that you have certain polymorphisms that you find within a subgroup of diseases. So a really strong relationship that maybe a lot of people know about is ankylosing spondylitis and the HLA-B27 mutation or variant that has a very, very strong association with the development of that disease. And so something like over 90% of persons with that disease have that mutation. And so it's suggestive that this is involved in the pathogenicity of that illness. And once you bring all of those people together in a multidisciplinary team meeting and you discuss those findings in the context of the phenotype and the presenting illness and maybe other aspects, then you're going to make a report and a diagnosis. And from there, you can identify possible treatments, potentially go to clinical trials, or you may actually find no association at that point in time. And that's one of the difficult things about this work, is that it's always evolving. Just because you can't see a relationship right now doesn't mean in a year's time there won't be that association between those variants you've initially discovered and the presenting condition. So you have to go back and review this work to make sure that that's actually still the case. So where I was trained, this is the workflow of how this process starts. You have individuals who present at the clinic, usually to a medical geneticist, and what they'll do is provide a suspected genetic disease diagnosis and order tests. And those test orders come to us, the clinical scientists, and then we do that process I just described, and we generate a report that takes on average about 2 weeks per person. Even when you have lots of people involved, it's because you're potentially, depending on the amount of genes you're looking at millions of genes. And you have to classify all of these, millions of variants, I should say. You have to classify all of these variants on their pathogenicity with respect to the condition. Once you do that, you generate a report, and then you sit down with all of the physicians involved, and then they go for genetic counseling if that's available. So how is this related to cancer? Because this is more of a broad level. So these are all the different structural variations within a genome that can lead to cancer, right? I spoke about some of them. So for example, repair mechanism disturbance, when you have sun damage or environmental damage that causes DNA to break apart, and then the repair of that gets introduces problems, and then that can develop cancer, like for example, skin cancer. And we want to look at all of these things and see how potentially, if there are particular mutations, stroke variants, that drive the development of this cancer. So you can take some genes from that tumor, and you can look and see what genes are being upregulated, expressed, what proteins are being expressed in large volume, what pathways are actually being pushed in this cancer. And you can say, okay, these genes in particular, because of these variants, are driving that cancer. And that can help actually to choose your therapy in a lot of cases. You can look at tumor evolution. So one of the things is unchecked cell growth. And because of that, you'll have mutations also introducing from parent cell to daughter cell. And you want to see how that tumor is evolving, because you could potentially predict how it's going to look genetically at a certain time point. And that will also tell you about the mutational processes within those cells. And so in certain cases, one of the strong associations with cancer and genetics is in hereditary cancer, right? So we can use genomic sequencing in establishing a predictive model for whether or not someone's going to develop that disease. And so you have here a case study where you'd have an individual patient, early age of disease onset. You have multiple primary cancers in the same organ, multiple primary cancers in a different organ perhaps, and maybe it's related to gender, maybe not. In a patient's family, you'd have 2 or more first-degree relatives on the same side of the family, cancer of the same site, cancer types belonging to a known hereditary cancer syndrome. So those things suggest a hereditary cancer, right? So the first thing when those people present to the clinic is you do a pedigree and you would see, are you picking out these patterns? And then you would want to look at not the cancer genome necessarily, but that individual's own DNA, because you want to see, is there a hereditary aspect involved in this? And so this can take the form of a multi-gene panel, oftentimes, and you group genes together to look into, because they're associated strongly with the development of the disease, of course. Now, panel science is quite a difficult thing, because you have to review a lot of literature and say, okay, these genes are involved, but not these ones, and then that may change over time. So it's a very fluid science. But you don't want to look at everything, because you could find things you're not looking for, and that can cause other ethical issues, right? So one of these things as well is you group panels on what are called high penetrance genes. So high penetrance genes are essentially, if you find a pathogenic mutation within a gene that's high penetrance, it's a high likelihood of you developing the disease, right? So in some cases with BRCA1 and 2, it can be upwards of 80%. And so that gives a good correlation between something predictive and perhaps taking intervention after the fact. And so this is some work published in Nature in Genomic Medicine, where they were looking at a cohort of 636 patients. And what they were looking at are all of the genes in those person's genomes that create proteins, right? And if you look at B, that circle there, that's a representation of the different types of mutations that are identified as pathogenic and likely pathogenic, right? So I spoke about some of those, like missense mutations and nonsense mutations, et cetera, right? You can see that there's a lot of pathogenicity, meaning that these mutations being present are going to lead to the development of that disease. In mutations that are start loss, so when the genome is being read, an enzyme has to come and bind and then start that process of making those amino acids. If it doesn't even start in the first place, you're not even going to have a protein, right? So it makes sense that you're going to have development of disease if there's no protein to do its normal function. And then you have a breakdown here of the most commonly mutated genes, which are shown ranked from bottom to top, right? So we recognize BRCA1 and 2, there's CHEK2 that's often involved. A lot of those are very, very classically associated genes with the development of certain cancers. So it's kind of no surprise, but it's interesting to see it like that. And then, of course, on this bar graph here is the fraction of pathogenic variants that were calculated for each cancer type. So you have mesothelioma, ovarian, and cervical cancer right up there. And again, it's a really good study because there's a lot of power. There are a lot of numbers in this cohort, but it's also telling us something that we really know. So what do we currently do? Well, currently at HSA, we send off all of our tests, just like the rest of the healthcare industry in Cayman. And we're really sending them off for these two tests. So Oncotype DX and MammaPrint in particular. And Oncotype is looking at breast reoccurrence score, right? And analyzes the group of genes that can affect how early stage breast cancer is likely to behave and respond to treatment. So this is not merely just looking to see the presence of variants and so on, but it's also to see how highly are these genes being expressed in this cancer. So these two tests look at the cancer itself and the genetic components of that cancer, whereas the middle one is looking at what is called your germline, which is your own DNA to assess your risk for the development of cancer. So it's more predictive, right? And that's BRCA1 and 2 in particular. And MammaPrint looks at an activity of 70 genes and then calculates a reoccurrence score that's either low or high risk. And so that leads me to where we're heading with this. What's the plan? So we're going to be providing local genomic analysis for the assessment of risk for the developing of certain types of cancer for the first time ever in the Cayman Islands. And that means that complex analyses will be provided faster, cheaper, and with greater local expertise than ever before. And we recognize that this is a whole healthcare effort, so we're working very closely with all the individual stakeholders across the island to deliver this. But this will mean that we won't have to send off our samples anymore, and we'll be able to interface with the physicians in greater detail, and so you can get more insight and a better healthcare outcome for patients. So thank you. Thank you very much.
Thanks. As we get set for the next speaker, there are a few cars that are blocking other vehicles. A white Toyota Mark X, 198563, 198563, a white Toyota Mark X, and a Honda Fit rental car, 189563, 189563. If that's your vehicle, can you please move it? You're blocking others.
Thank you. Good morning, everybody. And thank you to the organizers for putting this important forum together. And of course, I want to single out Brooke for making our slides very pretty. Jonathan, you did an excellent job, and I don't know if I can follow on you. So as we move in talking about genomic sequences, we talk also about the whole aspect of precision oncology or precision medicine. I have no real conflicts, but Jonathan spoke about the missense and the nonsense mutations. So if you see any misspelling, it's only there deliberately that you will understand the concept of missense, nonsense mutations. It's important for us to recognize that no two persons are the same. We're all unique, and so are our cancers. Cancer is multifactorial. I think Dr. Ravi earlier on mentioned something about people who have normal cholesterol. I said, but why did I get a heart attack? It's the same. You know, I don't smoke, I exercise, I eat well, I do everything right, and I'm still diagnosed with a cancer. Well, it's specifically because of the multifactorial nature of the genesis of cancer. And we can see a number of things there. And this is only a touch. There are several other things that are implicated in cancer: tobacco, chemical carcinogens, other environmental things, radiation, aging, obesity, diet. All of these have some form of implications. So therefore, we know cancer, like other chronic non-communicable diseases, they continue to pose challenges globally. People still present to healthcare with advanced cancer. So we know it's a challenge. And the whole aspect of genomic testing, which is important in terms of both diagnosis, prognostication, and so on, it's that many times it is not easily accessible depending on where we are or because of insurance coverage. And of course, when patients present extremely late, then you really question what may be the utility. And there, of course, the whole aspect of having precision treatment to ensure that once we have diagnosed the cancer, we know specifically what to use in that particular patient. So I will take us back. We remember the days when we were running around in the 1920s, correct? Those were the days when the main treatment option for cancer was really radiation therapy. And in those days, those radiation therapy were destructive, right? But by the early 1940s, there was introduction of chemotherapy. And even today, people are afraid of chemotherapy. But I can tell you that it has improved over the years. So at the turn of the century, we began looking at targeted treatments. And of course, the buzzword nowadays is immune therapy. Several patients come and say, Doc, put me on the immune therapy because somebody says that it is easier, it is lighter, and this and that. I know Dr. Google is always telling you about the different things. So what is important along that line? It is to recognize that there's no single silver bullet in terms of therapeutics, and we have to analyze each patient as he or she comes along so that we can strategically target that particular person's cancer. Just like how nowadays we have all these, when we see war happening, you can sit in a war room in country X and send off something in another country, right? So that is how we want to treat cancer, to be precise. Okay, I'm going to have just 2 short case discussions, and at the end of the day, I'm pretty certain you'll understand and say whether or not these 2 patients would benefit from genomic testing. One is woman XX. She's 43 years old, and she was diagnosed with bilateral breast cancer at the age of 35. She had bilateral mastectomy, and she completed the conventional chemotherapy, immune therapy, endocrine therapy based on the histopathology, what the pathologist reported on what was seen, and the immunohistochemistry. This woman has had multiple recurrences over the years between 35 and 43. And she has basically exhausted all lines of treatment. However, she continues to have very good performance status, and so therefore we know she has a will to live. Then our second patient is a young man, Mr. XY. He's 36 years old. He presented because he was just feeling weak and his energy was gone. He was diagnosed with iron deficiency anemia. He admitted that he was passing blood via rectum, which he thought was because he had hemorrhoids. He's a smoker, and he had colonoscopy, which showed polyps. Multiple polyps in the colon. He has a very strong family history of cancer. His father died at 45 from metastatic colon cancer. His uncle, his father's brother, currently has colon cancer. And his grandfather died of metastatic prostate cancer. Now, Jonathan mentioned that cancer is really a genomic condition. If we look at all those little small oval-shaped circles, we'll recognize that every aspect of cancer has to do with some form of genomic or genetic change. We look at cancer from a point of view from its genesis, from why is it that a patient who is doing very well on a particular treatment strategy today, and by the end of that 6 months of treatment, the cancer has suddenly progressed. It means that there has been a mechanism for resistance, and that resistance may be because there's been some mutation, even while that same cancer is going on. There might have been some kind of change, whether it's nonsense, missense, whatever sense it is, or anything of the sort like that. Good? I'm no better than Dr. Ravi. Good. And I think Jonathan went through this slide. There are about 4 different types of genetic or genomic changes that can go along, and I'm not going to elaborate on that. If not, Jonathan is not going to be happy. Good. So there's been a paradigm shift in cancer, and this is because of genomics. The important thing to recognize is that cancers may be diagnosed much earlier. With the use of genomics, there is the whole aspect of giving prognosis, the whole aspect of developing targeted treatments. Those are critical because you would want to know that once a cancer is identified, we know specifically what treatment strategy to use. And that's the whole aspect of genomic and precision oncology. Now, precision oncology, as we see there, we have precision medicine, precision oncology, precision this and precision that, right? It's really about ensuring that we make the right decisions at the right time about the right patient who has been diagnosed with a cancer, right? And of course, genomic sequencing holds the key to that. There are several different packages of genetic tests that can be done for different cancers. And we hear about next generation testing. Just Google it, and you get a whole barrage of different things. right? And it's about understanding what are these various genetic or genomic markers, what are those changes, those nonsense, missense, all those senses that can go along that are particular characteristic of certain cancers. It's important to also recognize that even with cancer A, you may find the same changes also in cancer B, right? And they're two different cancers. So sometimes some of these changes are not necessarily for one particular cancer, or even inherited things. So everybody knows about BRCA gene. BRCA, find it in breast, prostate, ovary. So you can go along and see what we're talking about. So genomic testing allows us to really, really target those particular drivers, those things we believe are really what make the cancer occur, right? And so therefore, targeting those drivers is really precision medicine. Within this, we talk about tumor biomarkers, and they talk about some of these proteins that may be expressed, whether they're in the cell itself, whether they're on the cell itself, whether they're in the cell membrane, whether they're on different organelles within the body. And some of these that are used nowadays include things like tumor mutational burden, which is looking at really the quantity of those changes that are on the tumor. And I'll demonstrate in a little while why that is important. And also what was spoken of earlier on about the instability, microsatellite instability, and the importance of understanding these, especially in cancer that has spread, we call it metastatic cancer, is that we can effectively use immune therapy to treat these conditions. Good. So I spoke about tumor mutational burden. It's just a quantity of those changes that are seen on the tumor. And of course, as we see there, immunotherapy plays an essential role in managing those patients who have a high burden, a high quantity of tumor mutations. Good. So we talk about microsatellite, and I'm going to attempt to use this. So we see on the left these nice gray blocks set up nicely here. These are all repetitive genetic fragments. But over here, in another language, we call it chaka-chaka, right? Over here, we can see them all over the place. It's really demonstrating the instability, right? But, of course, these are tumors that will respond to certain forms of immune treatment. If we go back to 2004, at the end of 2004, for lung cancer, there were only 2 noted genetic markers or genomic markers, right? But by the end of 2017, there were over 25. And 2007, may I remind us, it was about 5 to 6 years ago, eh? So by now, I'm pretty certain that there are many, many more markers. But if we look at the bottom of the column on the right, we still see that there are still a number of lung cancers that we're yet to determine what are the genomic or genetic changes that are seen. So, all in all, we clearly agree that it's all about understanding what is the cause, what's the driver. If you do not know the cause, then it makes it difficult to target treatment, right? That is all. And in the middle, we find genomic testing and genomic profiling, which is able to package that to us, and most times, or sometimes at least, point us to where we may be able to do precise treatment. Good. I spoke earlier about tumor mutational burden. Now, if we look in the red on the top, we see some different figures. But what that goes to show is that patients with high tumor mutational burden, and though they are treated with targeted treatment, they are more likely to respond better, right? We find that the response rate is really 3 times higher in those with high tumor burdens, and who receive targeted treatment as opposed to those who do not get targeted treatment. As we look at the middle, that looks at progression-free survival. That means that how long do patients remain in a kind of a remission status where the disease does not move on. And you see, again, it's about 4 times for those who are not. And on the extreme right, one piece of information that is very, very important. With some forms of targeted treatment, the overall survival has not yet been reached. So science and the role of genomic understanding has made that very, very important. So cancer is no longer a death sentence. It's just a phase in life that we need to address, like how we address hypertension and diabetes and so on. So, do we throw away conventional molecular diagnosis? Of course not. We cannot throw it away. And especially it depends on what our economic situation is. They continue to play important role. We're talking about immunohistochemistry, in situ hybridization, PCR, and I'm pretty sure everybody in Cayman Islands knows what PCR is, right? And Mr. PCR is, of course, to my left. So, but if you look at the red lines at the bottom, it tells you that with the conventional molecular diagnostics, there are still some gaps. And those gaps are simply because we miss some of those drivers. And so that's why we have to move on to the next phase of ensuring that we adopt the whole comprehensive genomic profiling to be a reality in today's clinical practice. It's not easy, but it is something that I think we should all move towards. Because having comprehensive genomic profiling gives most of the answers. And I said most of the answers because you'll find in another slide later on that it may not give every answer we're looking for today. So this slide, and I have no interest in Rush and Foundation Medicine 1, but it's important for us to recognize there that with using the next generation testing in Foundation Medicine, where either a piece of the cancer tissue is tested, that's the one in the orange, or from the blood, the blue and the red, where the genomic testing can be carried out looking for over 300 cancer genes, right? So it is a reality, and especially the one in the middle and to the right, where, for example, somebody has a cancer which is deep inside the abdomen or deep inside the chest. It would be an invasive procedure to be sticking a needle in to actually take out a little piece. It is not an easy walk in the park, but with a simple blood test, one will be able to look at some of these cancer DNA markers. Now, there are several cancers. If we look on the extreme left, we can see the names of many, many cancers: bone, kidney, liver, nervous system, prostate, cancer of unknown primary, pancreas, breast, and so on. All of these have some form of genomic change. But if we look to the extreme right, we also recognize that there are matching treatment options for these. So the whole aspect, the revolution of genomic medicine has given rise to precision oncology, which is very, very, very important. Because again, as I mentioned, we want to make sure that we treat the right patient with the right stuff at the right time. Lung cancer has been one of those that they have had a lot of research on. On the left, we can see the numbers of genomic changes. But a big proportion of lung cancer, 30% of them, have not demonstrated any particular change in their genome to date. I'm pretty certain that when I get a little bit older, there will be more of these. As was pointed out in my earlier slide, right? Now, if you look on the extreme right, we will see some different headings. And each of those headings refer to particular genomic changes and what are the different treatment strategies for lung cancer. Just to divert a little bit, some months ago, I had a patient who presented with a metastatic cancer. Wasn't sure where it was coming from, had a liver biopsy, and then my dear pathologist said, this looked like lung cancer. So, okay, fine. Sent it off for genomic testing. And then I got a call from a lab somewhere in the States saying, your patient have a RET positive cancer. I'm going to send and tell you which drug you should use. But it was very exciting because that particular type of genetic mutation or genomic mutation is not regularly seen. And to treat that, it's a specific type of drug that has to be used. We all know about breast cancer, and everybody of us here today has known somebody or have a family member who has had breast cancer. We know these ones that are hormone positive, hormone negative, or whether or not they carry the bad HER2 marker. Breast cancer, as well, as we can see there in the donut, carries a number of mutations, right? One of the frequent, I think Jonathan mentioned the whole thing about the BRCA mutation. This is what is inherited. And of course, I mentioned earlier on, BRCA mutation not only has implication for breast cancer, but also have implications for other cancers, including pancreas, prostate, right? The BRCA gene, we have two BRCA genes. One is on chromosome 17 and one is on chromosome 13, as you will see there. Colorectal cancer, same story. There are a number of mutations, and yes, we go beyond just sending samples, patients with early breast cancers, where Jonathan mentioned, we send them off to look at specific mutations that may allow us not to, or tell us if that person will benefit from chemotherapy. It's the very same way for our colorectal cancers. We do send off and we get different mutation analysis, which guides us as to what therapy to use. The other thing which I find particularly interesting is, of course, prostate cancer. And you might ask, why have I gone through these specific cancers? Because they're the most frequent ones that we treat every day here in the Cayman Islands. And prostate cancer is now becoming the order of the day that somebody who presents with prostate cancer, we should probably do some form of genomic testing, because that become the order of the day. Right? And as you can see in the red, one of the biggest mutations we find is actually BRCA. Right? Now, there are implications for that, and I think if those who were here last night would recognize that what do we do with some of these analyses when we get them? There are certain ethical implications for doing genomic testing, but that's for another day. So I put this slide there because I want us to recognize this is not just, sporadic ideas. There have been several research that are carried on. This is just one of the research looking at, again, providing the evidence to demonstrate that when these genetic or genomic changes are seen in the various cancers and they have targeted treatment, then the survival and possible remission is much, much higher than those who do not get targeted treatment. And I mentioned earlier the aspect of using blood to search for some of these cancers. Cancer is found anywhere in the body. And, there's a good senior person every morning on the rounds. He says, life is in the blood. He shall remain nameless. But what happens is that the blood and the lymph bathes every single tissue in the body. And in doing so, the cancer, although it may be at position X, but by bathing the cancer, then either the cancer cells themselves or cancer cells that are pegged onto other cells, whether they're pegged onto platelets or white cells, those are washed into the bloodstream, and that's the reason why it's so important and easy to use. Easy to use those DNA samples from the blood to look for it. Advances have been much more beyond the blood, and of course we'll see in other aspects, for example, using buccal swabs, one may be able to diagnose certain genomic markers that may be related to some cancers. And certainly, for example, even in colon cancer, the use of stool sampling to look for certain changes. So genomic testing and cancer diagnosis has been evolving. Now, since you asked me about those two cases that I presented, I will give you the answer. Case XX, XX means female, by the way. So case XX, she had metastatic cancer, good performance status, but we're near to the end of the road in terms of options. I definitely believe that she would benefit. Case XY, of course, XY is male, not yet diagnosed with a cancer, but he has a very, very strong family history. And certainly we know that we have hereditary colon cancer. I mean, it's well known. And so it is important that we do genomic study in this gentleman, just to, first of all, to find out, and that will guide what kind of therapeutic options. So, for example, and I'm delving now into the field of the surgeons, so I declare I may be wrong, but this person, this young man, he has a lot of polyps in the colon, and one of the things that may be done is that given the strong family history that he has, and if he has these genomic alteration, maybe preemptively, it may be considered that the surgeon could remove that part of the colon that has all of these polyps. I declare I'm not a surgeon. Good. So, benefit. Does everybody benefit? No. But there are people who benefit from it, and I mentioned two different types of persons who may benefit from it. Right? It is important along the line when even sometimes you have some cancers, cancer of unknown primary. Don't know. You know it's a cancer, but you don't know where it's coming from. And so the genomic markers may be able to help us, to give us prognosis, to guide us for treatment. Those are important people. We encounter some very aggressive breast cancers, like triple-negative breast cancer. And these persons may benefit from it in terms of trying to identify different markers that may be able to predict what kind of treatment strategy we may give. Of course, you mentioned the whole aspect of people who have been through rounds and rounds and rounds of treatment, but yet they still have the cancer. Then we will have, then it behoves on us to really do genomic testing to see what else is there and how can we target them. So this is an example of a patient with an ampullary adenocarcinoma who genomic testing was performed on. Now, this is the gene, and I'm certain Jonathan would understand it. I don't. It's just some nonsense to me, right? But identified here, if you notice here, that there were no known therapeutic or treatment interventions for this particular type of cancer with this particular change. However, what is there is that for other types of cancers with this particular marker, they have used these treatments before. And so it is now a time for when the physician have that discussion with the patient and say, listen, this may not be for this particular one, but it has been used with benefit in that, and so on and so forth. So there are times when you need that kind of discussion. Now, this is a person with colon cancer, right? And the person definitely have a treatable target, a BRAF mutation. And we know regorafenib can be used there. Good? But in the case that we don't have regorafenib, then these are other options that may be used. It does give us options. And of course, those who want to hear more about immune therapy, you know, with the KRAS wild type and that kind of thing. But if I continue like that, then in some jurisdiction, they'll be saying that I'm using bad language. Now, there are challenges. There are challenges. And I mentioned last night from our opening speaker from the UK Ambassador for Health last night, who spoke about the ethics of it, right? The question is, so you find a mutation so mommy has a BRCA gene. What are you going to do about mommy's daughter who is 5 years old, who is 10 years old? What are the implications? So it's not just a little willy-nilly thing, right? And of course, it's not appropriate for everybody. And it's not a silver key or a golden key to make sure that everybody with a cancer will live longer. Unfortunately, that's not the case of it today, right? One may have a genomic mutation, but yet the results do not give us the idea for a specific type of treatment strategy, right? Sometimes some of these mutations are even poorly understood by those who do the test, but for us physicians, complete nonsense, right? Only a subset of these cancers really the treatment of them benefit from these. So we still have a far way to go. Managing big data. Jonathan spoke about pulling all this data together. Managing big data is also a challenge. The high costs, because some of these things run into the thousands. And the elephant in the room is really about research, and especially among Black people. Not enough research is done. This is a patient with a myeloid sarcoma, and a mutation has been identified. And as I mentioned before, no therapeutic option, not even a second option. But yet, we have identified it, and that's one of the drawbacks that can occur. So there's a whole revolution, an ongoing change in the field of genomics. There is the whole aspect of the genomic profiling. It's managing big data. It's managing all of this evidence that's coming about, and then all the licensing authority are pushed or forced to approve a number of different things, right? And we now have everything at our fingertips, and we can touch everything and we get the answers. This is what is happening, and it's about how do we get there? How do we manage all of this? So that is just a snip of precision oncology, and we try to do now what patients need next. Thank you.